Okay, I'm going to go ahead and start recording. I'd like it if you all were quiet. You've had about 45 minutes or so to catch up with any of the assignments you missed. Some of you wasted that time, but that's fine. We all learn from our mistakes. So I'm going to go through this chi-square statistical analysis. Then I'd like to go through the actual lab to make sure we all understand what's going on. As Owen mentioned earlier, the formulas will be given on the AP test. So they'll give you this and they'll give you a data table. Let me insert that here. So you'll find these data tables anywhere on the internet. I want to try to look at this again. It's not incredibly important that you can still do this if you don't understand what I'm about to say. And they're going to format these tables slightly differently, but they're pretty much all going to be the same. These are p-values at the top. So this is a probability of that happening. We pick 0.05 as a significant p-value. Again, as I discussed yesterday, as we drew on the board, when you look at the data, at any bell curve data, and if it's a natural phenomenon, it generally happens in a bell curve. We worry about these extremes, these tails, because we call it tails because it looks like a tail, right? Yes. So we worry about these tails because they give us this information. And so if we set this value, the max here at 0.05, then and here obviously, actually it's not really 0.05. We only worry about this half, so let's just, because it would be Oh, I see. Sorry, I have the circle real small. So it really would be 0.025 on this end and 0.025 on this end, because it's 0.5 divided by 2. But anyways, forgetting the numbers themselves, we usually don't worry about the negative side. We just worry about the right side of the tail. So if we just look at just this half and just concern ourselves here, this is our limit. We say that anything that's on this side of the bell curve, we're in between the two tails, we're going to accept as possibly happening by luck. Something else occurred, but whatever the issues, we call it statistical noise. Or people call it statistical noise, not we. I don't call it anything. But this is any data points that are found here along the major, along the mean, near and up the mean. We say this part, which is equal to 95% of the data. 95% of the data is within this white part of the bell curve. But in this red zone is, again, 0.025 or 2.5% of the data is over here, and 2.5% in the other tail is extreme. So then if we find the data point over here, then we kind of consider it to be significantly different from the average. Significantly different from what It's unlikely that it's accidental. There's something weird going on. It's not explaining anything. It's just saying that statistically there's a significant difference. So, when you look at chi-square now, look at these tables. What are these values? Well, this is the 0.05, so that would be, half of it on the right end, the right tail, half of it on the left tail. This is this is a table value of 19.25. We call it a critical value. A critical value. So this is a critical value. This is known as a p-value. So the p-value, and we call specifically this value of 0.05. I'm going to highlight it, I'm going to underline, I'm going to circle it, and I'm going to label it as being the alpha value. All right, because it is alpha. It is the one that most people, like any one that you choose, you can choose alpha. You might choose alpha in the lab that you're doing research at 0.05, which would mean what? That it's all the way out here. Like it's the very tip of the tail. No, what am I saying? That's the same, isn't it? Sorry. You might choose 0.001, which is a lot less than 0.005, right? Oops. I don't know how that happened. I can't undo it. Sorry. Maybe I can reinsert the photo. So the bottom line is, when we look at this data, 0.0. Oh, I see. I see what's going on. These are different degrees of freedom. At 0.05, I can choose. I was confused on why there's two 0.05s on there. Why can't I write on it? What's going on? Is this thing punking me right now? 0.05, that's this, where you divide it into half, because half's on the right and half's on the left. But if I was going to join 0.001, that's all the way out here, at the very tip of this red tail. When might you choose that? If you're absolutely sure about your data, if there's like very, 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 just that much of a difference from the expected value, you would consider it significant. For instance, if I threw something up and it kept going up into space, I would consider that a very, like, just if it happens once, it's a big deal to me, right? Versus, I measured, it's 1.01 versus 1 gram. Yes. Say what up? Not really. It's just the end on the negative end of the lower end. This is the high end on this end. So any if you think about a grade, because that's the easiest way to think of it. If you think about this as being the A's over here, then these would be the F's, right? And so what are the chances of getting an A? 90% A. It's possible. What a chance to get 100% A on a difficult test? Less likely, but still possible. So you have this tail end over here, and the same thing with the F's. What are the chances of getting a 50% F? It's possible. What are the chances of getting a 0% F? Really unlikely. In fact, I would argue it's highly unlikely to get a 0% F unless you're trying. If somebody on a difficult exam, a multiple choice exam, it's statistically very rare to get a 0%. Of course, unless it's cheating. But in any case, the point is, when we're looking at these values and we look at this chi-square, what is a chi-square is, we're going to compare the critical value, and we're going to say, let me see. If our chi-square, if we're looking at 0.05 with 1 degree of freedom, what would our critical value be? 0.05 with 1 degree of freedom. 3.84. What if our number ended up at 4%? We reject what? At 4. So it's higher. So what does that mean if you think about it? And this is why they had you in the activity write down the p-value range. If you have a number, this is a test number. This is not a p-value, right? This is a test number. So if the test number is 4, that means it's here somewhere, right? So it's in between. It's in between 0.5 and 0.01. So it's actually, where would that be on this graph? It would be over here. It would be to the right of the critical value. So that any number bigger, any chi-square bigger than your critical value is gonna mean that your p-value, the chances are less that it's just by chance. Because look, the chance you're looking at a value of chi-square here that would mean there's a 5% chance there's a 5% probability it's just chance. If you have one of 4, then it's closer to 1%. That's a lot less, right? So it's a lot less likely. So we reject the fact that it's a null, that there is no difference. We say that is a significant difference. Yeah, question?
So you reject the null hypothesis even if it's bigger and not only like, at first I thought it was only if it was smaller, but we rejected it too if it's also too big.
No, if it's bigger, not smaller. If it's smaller, we accept the null hypothesis. Because the opposite is true. Let me Let me erase this. Let's say, I'm sorry, I have a headache, my nose is stuffed up, my nose is running. It's really miserable. I'm really miserable right now. So let's assume for a second that we're doing an experiment with that gravity experiment, and we're looking if there's even a 1% chance, less than a 1% chance. I'm not going to accept anything that is, my limit is going to be 1%.
Your p-value is 1%.
My p-value is 1%. The specific p-value that I just circled would be called the alpha. I'm going to compare it to this alpha. I'm going to say if my chi-square is larger than 10, then I reject the null hypothesis. But if it's smaller than 10.828, I'm going to accept the null hypothesis. So let's say my chi-square value is 6. That means it's in between 0.001 or 1% and 5%, closer to 5%. I'm going to say no. I think it's likely there's a good possibility that it's due to random luck. It has to happen at 1% or less. So that means my chi-square value has to be greater than 10.828 in order to reject the null hypothesis. Let's go back and look at the null hypothesis. That's why I define the null hypothesis in your chi-square discussion in your summary. So what is the null hypothesis? Let's remember, the null hypothesis, go ahead, is the assumption that what?
Status quo. It's due to chance.
And it's due to chance. That any difference from the expected value is due to chance. And what does that mean? That doesn't necessarily mean error. That could mean that anytime you make a measurement, things go wrong. Their rulers are not perfect. Rulers can be off by a fraction of a millimeter. Balances could be off. Breezes can impact. Someone could sneeze. There's random things that happen all the time. And there's if if the measurement comes within this bell curve, within this limit that I lay out, again, what is my limit? It all depends on what you're measuring. It all depends on what you're measuring. Yeah.
Does this contribute to why we can't prove anything? Because
Yes, that's why. This is all trying to deal with it. It doesn't really contribute to it. What it does is it's our way of trying to deal with the fact that we can't prove it. Like there's no way for us to prove it. So how do we get to the point? How do you and I agree that this is significant? Like how do we agree? We have to do. All right. So let me give you an example. MS, multiple sclerosis. You know what this is? It's a demyelination of the nervous fibers. Your body is an autoimmune disease. Your body starts attacking your nerves and starts to eat your nerves up. It starts sometimes in the brain, sometimes in the periphery. My brother had it. He died from it. And what it does is it starts to eat away. Your body's white blood cells start to destroy the myelin of the nerve system, and you start to lose function. And ultimately, it kills you. So it can kill you. It doesn't always. And other things might kill you later. It depends. Anyways, MS is a serious disease. Researchers found that using statistical analysis, found that almost everyone that had MS, it could be traced to herpes simplex 1, infections. So people that have it, that doesn't mean there's a causal relationship. It means that it's likely HPV is the first step to getting MS. It causes MS, yes, but it doesn't. Not everybody that gets herpes simplex 1 gets MS. There's other factors, but we know that it's certainly a causal factor. How do we know? We looked at the data and we compared it to what we expected, and it's obvious. It's a tremendously well accepted fact that because the study was so large, because we had such good statistical data, very few people really argue about it. Now, are people still arguing? Sure. Because you can always find another. I mean, my gosh, you guys know, you guys watch the news, you know humans can argue about everything. Is there anything we can disagree on? I mean, seriously. So we have, in science, we have to seriously take a look at how we can come up with some standards where we can just accept that this is the case until we find something else that kicks that rule out. Because otherwise, we have no conversation we can have. We can't have any agreement. Yeah.
So, like, rejecting the null hypothesis is like the first step in accepting that something is significant?
Yeah, it is. In fact, rejecting the null hypothesis by definition means that the difference is significant, and you accept.
No, and it's not due to chance.
When you reject, I don't know why I put a G. When you reject the null hypothesis, you means you accept the alternative, which means there is a significant difference. Either there is or there isn't. To accept the null hypothesis means that your data, whatever that measurement you've decided is within this area here, within the bell curve, it's well within acceptable margins of error. I'm not even going to take it seriously. If it's extreme enough, then I'm going to say, hey, I'm sorry. That's important. This is significant. I'm going to say there's something either wrong with the theory or if your idea that this. So what's a good example of how we can use chi-square in genetics when we're looking at evolution? Remember the 9th grade, we talked about evolution, and we talked about, oh my gosh, what's this? Oh my gosh, I'm blanking out here.
the table that's like
It's a postulate, oh my God, I mean, what is it? Just you saying table clicked Hardy Weinberg. Why? I don't know. So Hardy Weinberg says that, look, if these 5 things are not occurring, then no evolution is occurring. No statistical. So But if you have a statistically significant difference from the expected value of traits, of alleles, then you have to say evolution's happening. All right, so so remember, evolution is when your allele frequency, right, changes. That's it. You have evolution. We're not talking about some ape turning into human. It's not like, some kind of sci-fi movie. We're saying the allele frequency in the population's changed. Can you add up those allele frequencies and end up with a different species? Yes, according to evolution you can, but that's not the key. The key here is that evolution changes. The theory of evolution says if the allele frequency in the population changes, you have evolution, period. Hardy Weinberg says that p squared plus q squared equals 1, which means they have to be equal. They also say, what does it say?
Table.
Yeah, well, you can derive a lot of understanding from this, but the point is, if you find a frequency, remember that p squared is the dominant homozygous, and this is the the recessive homozygous. With a little bit of calculus, you can find that P plus 2PQ plus Q equals 1. So now, using those two realities, if you find out that the homozygous recessives are greater than you expected, then you can say, if it's significantly greater, you will see differences depending on your sample size. So is that difference significant enough for you to decide something big is going on, or is it within that bell curve where it's like, well, you know what? It's within the bell curve. That's just a measurement error. That's a statistical noise. That's me miscounting. Like, that could be a lot of different things that are going on. But if it's enough, if you have enough of this difference is big enough, well, how are you going to decide what's enough, what's big enough? You apply the theory to your numbers, then you apply the numbers to chi-square, and you follow that chi-square procedure, and that gives you a conversation starting point where two scientists can sit down and say, well, gee, the data really shows us that something big's going on here. I think we should think about publishing. Yeah. Well, we got 2 minutes left. Does anybody have any questions about chi-square? How to do it? The steps are written in the review, in this AP Biology. If you need any help, I'll be here after the meeting until 4. I think the meeting's over before then, but we'll see. We're not leaving till the bell rings. Don't even go near the door, please. You guys give me the hives. And put the chairs up. Tomorrow's a practice FRQ. Yes, ma'am. You'll be working in teams.